Um, so, so first of all, thanks Dennis for the invitation and thanks for organizing this online seminar. So my work focuses on neuroimaging genetics, which is the joint model that's brain imaging and genetic data. So I will be talking about some of our recent work where we adapted some of the well-developed statistical genetics methods, in particular heritability and genetic correlation analysis, to the context of neuroimaging genetics and to handle some of the challenge there, such as large sample size or very large number of phenotypes. So I think many people are familiar with the concept of heritability, which measures the proportional phenotypic variation due to additive genetics if it's narrow sense heritability. And now with the increasing availability of genome-wide data, we are now also able to measure the so-called SNP heritability, which captures the phenotypic variation that can be explained by common genetic variation across the genome. So the most classical model for SNP heritability estimation is, I think, this linear mixed effect model proposed by the GCTA paper in 2010. So it basically partitions the phenotypic variation into the genetic variation and some environmental factors. And the genetic effects is assumed to be a linear additive combination of the individual SNP effects. And the effect size of each SNP is assumed to be a Gaussian distribution with zero mean and some common variance. So this gives a linear mixed effect model. And if you compute the covariance structure of the phenotype, it can be partitioned into two parts. The first part is related to the genetics, which is the total genetic variation times this empirical genetic similarity matrix, which measures the genetic similarity between pairs of individuals in the data. And the second part is related to some environmental factors or residuals. So over the past several years, there are many extensions to this basic model. For example, we can have different ways to quantify the empirical genetic similarity between individuals in contrast to this simple linear correlation of the genotype. And we can also, for example, stratify the SNPs based on their minor allele frequencies or linkage disequilibrium scores or functional annotations and incorporate multiple genetic similarity matrices in the model to partition the phenotypic variation into the different sets of SNPs. But to just keep things simple, I will just focus on this basic infinitesimal model in this talk. There are two general classes of statistical methods that can be used to fit this model and produce a heritability estimate. So the first class will be the likelihood-based methods, which is also the classical method used in the GCTA approach. So to use this type of method, we need to further impose some distribution assumptions on the phenotype and genetic effects. So normally we assume they are all normal distributed. So you have a fully specified probabilistic model. You can calculate the likelihood of the model and then use some iterative optimization algorithm to maximize the likelihood and then produce the parameter estimates and heritability estimate. So we know this method is statistically efficient for quantitative traits and when these distributional assumptions are all met in real data. But we also know there are some weakness for this method. For example, when we analyze those ascertained case control data set, this method would produce biased estimates because ascertainment would makes these genetic effects non-Gaussian distributed and also introduces some G-by interactions in the data. So these distributional assumptions are violated in this case. So you get some biased estimates. There's a second class of statistical methods, or the moment matching methods, that can be used to estimate the heritability. And this class of methods includes very popular LD score regression, which doesn't require the individual genotype data, but only needs the GWAS summary statistics plus some external reference panel to estimate the linkage disequilibrium within SNPs. So it's highly flexible and particularly useful to combine results across different cohorts without sharing the actual individual level genotype data. But if we do have access to the individual genotype, then we can also use this Huysman-Elston regression, which essentially regresses the empirical phenotypic correlation onto the genetic similarity to produce a heritability estimate. So both LD score regression and Huysman-Elston regression are moment matching methods, meaning that they don't assume these phenotype and genetic effects are Gaussian distributed, but in contrast, they just match the first two moments, the mean and variance of the data. So they are more computationally efficient relative to these likelihood-based methods because they are non-iterative. So in this talk, I would just apply these moment matching methods and leverage their computational efficiency to address some large-scale heritability analysis in practice. So just to summarize, in heritability analysis, if you have the GWAS summary stats, you can do LD score regression, and you can have a closed form estimator, which is the average of the chi-square statistics across the genome minus 1 times some constant. And if you have individual genotype data, you can do LD score regression, so which regresses the phenotypic similarity onto the genetic similarity. And you also have a closed form estimator. So I put a third case here. So if you have both the GWAS summary statistics and the individual level genotype data, so the heritability estimation can be formulated as a polygenic score type of analysis. So you take the effect sizes estimated by the GWAS and use the effect size to weight the genotype and do this weighted average. This produces a polygenic score. And you do this inner product between the polygenic score and the phenotype and properly shift and scale this inner product. And this also gives a heritability estimate. And we can show that all these three moment matching based heritability estimators are actually statistically equivalent. So depending on what data types you have, you can choose the most convenient estimator. The third representation may seem to be very useful because it requires both types of data. But in the next few slides, when we generalize this to genetic correlation analysis, we can see that this representation can be useful in high-dimensional genetic correlation analysis. And here it's just a conceptualization. So we leverage this computational efficiency of this moment matching methods to some large-scale heritability analysis. And the first example is the UK Biobank analysis. So the challenge in UK Biobank is that you have a very large sample size and often hundreds of thousands of subjects. So the traditional GCTA methods can be difficult to apply because the complexity of the likelihood-based algorithm scales cubically with the sample size. So moment matching methods would be much more computationally efficient to meet this computational challenge. But we also have the second challenge that even with moment matching methods, the genetic similarity matrix is huge. Say you analyze 100,000 subjects, then the genetic similarity matrix will be 100,000 by 100,000. So it will be even just difficult to read this huge matrix into the memory. So we further developed so we have some computational tricks where we can iteratively read columns of the genetic similarity matrix and compute some statistics on the fly, and eventually combine these statistics to produce a moment matching estimator. So even if you only have a single desktop, this analysis can be performed within minutes. So we apply this method to over 550 traits in the UK Biobank and estimate their heritability. We also looked at how these heritability estimates can be moderated by some major demographic factors such as age and sex and socioeconomic status. And we found some of these traits have significantly different heritability in males and females. For example, these quantitative blood pressure measurements and self-reported high blood pressure or the hypertension disease codes, and all these traits give consistent results that the heritability of these blood pressure related traits in females is significantly higher than in males. We also found that the heritability of a majority of physical traits such as height and BMI decreases as the age increases. And this might be due to some accumulated environmental factors on variation of this phenotype across the lifespan. So this is the first example where we use the moment matching methods to deal with the large sample sizing in heritability estimation. As a second example, so UK Biobank also collects multimodal brain imaging data for thousands of participants. And the challenge here is that relative to the analysis I just described, the sample size here is smaller, it's moderate. So we have around 8,000 subjects here, but the number of phenotypes can be huge. For example, because of these three-dimensional high-resolution brain images, we can compute the cortical thickness at each and every location on cerebral cortex. And this will give us hundreds of thousands of cortical thickness measurements across the two hemispheres. So moment matching methods can be used to address this very high-dimensional problem. And here we show this high-resolution surface maps for the heritability of cortical thickness measurements. And it can be seen that for a lot of brain regions, the heritability of cortical thickness is moderately heritable. So the moment matching methods can also be generalized to some genetic correlation and co-heritability analysis. So if you have two traits and both traits have GWAS summary statistics, we can run LD score regression, which is basically the average of the product of the statistics across the genome, corrected by a bias term, which depends on the sample overlap of these two traits, and then scaled by a proper constant. So if you have individual genotype data for both traits, we can do Haseman-Elston regression and also have this closed form co-heritability estimator. And there's a third case. If you have the actual genotype data for one trait and a GWAS summary stats for the other trait, we also have this polygenic risk score type of representation, where we take the effect sizes estimating the GWAS of the second trait to weight the genotype of the first trait. And this weighted average will give a polygenic score. And we do an inner product between this polygenic score and the first trait, and then properly shift and scale this inner product. And this also gives a co-heritability estimate. And we can also show that these three co-heritability estimators are statistically equivalent. So depending on what data types you have, you can choose the most appropriate, most computationally efficient estimator. The third case might not be very useful if you only have a small number of phenotypes to analyze, because you can simply run GWAS for each of these traits and then run LD score regression. But this can be very useful in neuroimaging statistics, where you have a large number of neuroimaging features, and you want to compute the genetic correlation between each of these neuroimaging features and a cognitive or behavioral or disease trace that has GWAS summary statistics. So in this case, if you want to do LD score regression, you need to run like hundreds of thousands of GWAS to get the summary stats, which might not be computationally feasible. But with this third representation, you only need to compute a polygenic score and then correlate this polygenic score with the phenotype, which can be highly computationally efficient. So as an example, we did this analysis in the UK Biobank, where we estimate the genetic correlation between these fine-grained cortical thickness measurements and education. So we have hundreds of thousands of cortical thickness measurements here, and to map the genetic correlation with education, we did this GWAS of education attainment in the UK Biobank and get this GWAS summary stats. And then we use the GWAS summary stats to create a polygenic score and correlate the polygenic score with this massive number of cortical thickness measurements. So this high resolution genetic correlation map shows that there's strong genetic correlation between the cortical thickness and education in these motor areas, and also some superior temporal cortex, which involve some language regions in the brain. We also repeated the same analysis with the verbal logic reasoning UK Biobank. So this is a phenotype that is highly genetically correlated with education, but it has a smaller sample size in the UK Biobank. So it's less statistically powerful, but we generally see the same pattern where the motor regions and superior temporal cortex show strong genetic correlation. So this might suggest that these morphological features in these brain regions might share some genetic origins with the cognitive ability. So I guess I'll just stop here, and thanks for listening, and I want to thank all my collaborators.